And now it is my pleasure to introduce Mr. Earle T. Holsapple, who is the Chief Executive Officer of SciTech. Earle.
Thank you very much, Brian. It's a pleasure to be presenting with you. Brian, for those of you that don't know it, is SciTech's Chief Medical Officer and my partner in this development effort. We're going to talk to you today about our breakthrough nanoparticle drug delivery platform that enabled Fenretinide, the anti-cancer agent that for 35 years has shown such great potential, but didn't achieve that potential because of its hydrophobic nature, so that the drug could never be delivered consistently to the cells. But we're not going to talk about that today. We're going to talk about nanoFenretinide's use in the treatment of COVID-19. So you're looking at a cell that is surrounded by coronavirus, and we want to talk to you about what Fenretinide is going to do to those cells and to the virus. First of all, I'd like to talk to you about a company overview. So you'll recall from previous presentations that we've made to the oncology groups that Fenretinide has been around for decades, shown great potential, first developed by J&J as a dermatology product and a breast cancer prevention product, and just never achieved what it was supposed to. It was then handed over by J&J to the National Cancer Institute. They developed it into a therapeutic, but again, due to its hydrophobic nature, again, didn't achieve its potential. But we solved that problem. We're going to talk about that and how it applies to COVID-19 as well. We were anticipating running a phase 1/2 clinical trial at Rush University Medical Center about now. Unfortunately, due to the pandemic's arrival, clinical trials in oncology were put on hold, and we took a right-hand turn, so to speak, because we realized that our drug has very significant potential as a therapeutic in treating SARS-CoV-2. So we're going to talk to you about that, the clinical trial we're about to embark upon, and the healthcare systems where we'll be running it. Let's just do a refresher on what ST-001 is and where it came from. So it's a nanoparticle drug suspension product intravenously administered. The nanoparticles look like this picture on the left of a miniature onion with the Fenretinide encapsulated between the layers of phospholipids or fatty substances, protecting the drug from water and allowing it to be distributed through the circulatory system in sufficient quantities to reach the cancer cells, or in this case, the cells that are infected by COVID-19. But it's unique in the sense that we can deliver high doses quickly and overcome the shortcoming of Fenretinide. We're here to talk to you not about all of these projects, but the last one, COVID-19. We're just about to start the non-Hodgkin's study. As I said, that was $7.5 million, to be closely followed by the small cell lung cancer or breast study. But today, we want to talk about a 6-month plan to bring ST-001 into the market for the treatment of COVID-19. We need $1.3 million now, an additional $4 million for phase 2 to have this product ready. And that's why we want to talk to you. Why invest now? Because ST-001, Fenretinide, generates antiviral activity, diminishing viremia and reversing the effects of the infection. Its anti-inflammatory capacity reduces the immune response, where you're getting an overreaction to the virus infection. It also impacts oxidative stress and cytokine storms. The combination of those direct and indirect mechanisms of action will result in protecting from organ failure, lung damage, and rescue patients from having to ever get on a ventilator, which is nearly the kiss of death these days. The study is carefully designed to demonstrate the safety of Fenretinide in an acute setting where it can be infused quickly so that there can be a reversal of the antiviral or inflammation activity right away. pronged combination to be used much like in HIV, two or three different drugs in combination to treat the disease. The market after COVID-19 is huge because it has potential in treating other viruses, but obviously, most importantly, the cancer indications that we've been talking about. So we're here to talk to you about do well while doing good. And it's been fun to make that right-hand turn. Why? Why is it important that we did so? We know that Fenretinide historically has shown antiviral activity against dengue, Zika, West Nile, and other what they call flaviviruses. We didn't have confirmation that it worked in SARS or MERS. Therefore, we sent our drug, ST-001 and Fenretinide, had them both independently tested to see if there was activity against SARS, most importantly. And the good news was there was. This was done at Utah State University's virology laboratory. At the same time, there was a South Korean study where they tested a whole range of about 1,600 drugs worldwide to see how they would impact the treatment of MERS. And Fenretinide was one of 50 drugs showing the greatest potential to reduce the impact of MERS. So these two data points make it clear that Fenretinide ST-001 have great potential in the treatment of COVID-19. The literature also makes the case that this multi-clinical opportunity gives us numerous at-bats. It's not just one mechanism that will drive this success. This impacts virus life cycle and side effects, including cytokine storm, lung damage, and viral pneumonia, as I previously stated. It's IND ready, basically, because we were prepared to enter the clinical trials at Rush University about now. We had an IND in hand that we received in December. We were ready to go, basically. Now we can use that same data to take advantage of the coronavirus treatment acceleration program at the FDA to get rapid approval for use of this drug in that clinical trial. So let's go back and recall what Fenretinide, the API, is. It's a synthetic vitamin A analog retinoid anti-cancer agent that exceeds the capabilities of other retinoids that have shown so much promise but never achieved their potential, primarily in a lot of cases because they were either hydrophobic or too toxic. J&J developed it, as I said earlier, tested it in more than 3,000 patients in numerous diseases over many years, and the worst side effect was night blindness, so it's a safe drug. The NCI took it over from J&J, tried to convert it into a therapeutic agent, but failed in their endeavor because of the same bioavailability problem that had been hindering Fenretinide for decades, a problem that we overcame with that delivery system that we just reviewed. So why is this mechanism of action so important for the treatment of COVID-19? Let's look at the left side of this slide. What's the virus doing? It's creating viremia, which results in massive infection. It increases interleukins 6 and 22 and others that causes all sorts of side effects, inflammation and organ failure and so on, and also extracellular hem, which is not good. Coming at this from the right side of the slide, ST-001 in its 4-hour infusion, administered at a very low dose relative to what's already proven could be administered using an intravenous delivery system in the case of T-cell lymphoma, is going to inhibit viral replication, suppress inflammation and cytokine storm, and all the side effects that we've talked about while protecting the alveolar in the lungs and preventing damage and allowing oxygen and blood to come together for circulation through the body. Published biomarkers for COVID-19. Look at this list. Brian, I think, had already shown you this, but I took those in blue to make the point that these are biomarkers that have been mediated by Fenretinide. So we know, we have very, very strong opinion, shall we say, that we will suppress IL-1, IL-6, and IL-22 minimally. Those three in combination should reverse inflammation, should reverse the infection, and should result in the patient recovering. The point is, however, that of all the biomarkers, Fenretinide has had an impact on over a third of them. So that gives us lots of at-bats, and only conducting this study will we know for sure. Our 6-month plan to have this drug ready, by the end of the year is really critical because of the expected second wave or increase in the first wave. We've got a phase 1 trial ready to go to confirm the safety of the formulation in this treatment, and also to arrive at a dose effective for treating this disease. Then move on to a phase 2 to confirm the efficacy, while at the same time collecting data on the in vitro activity or the biomarkers to confirm the hypotheses that we've been making. At the same time, we're making the drug. We're scaling the drug manufacturing at several different sites, so that at the end of the phase 2, assuming that we'll receive emergency approval from the FDA, we'll be ready to go into the clinic, in a commercial setting in January. We have a very, very detailed phase 1a/b study. I don't expect you to read this. I just wanted you to see the immense amount of detail that Dr. Leyland-Jones and Ralph Parchment put into the design of this therapeutic approach, taking into consideration all the possible impacts from various biomarkers and so on. So it's an elegantly designed study. This is going to cost $5.26 million for phase 1, 2, and scaling of the manufacturing if we get that far. We will probably know whether it will work or not by spending about $1.3 million. So we're looking for that right now. We have a regulatory plan that's in place that's really aggressive. We have the IND for the oncology setting, where we filed a pre-IND for the COVID trial. We will cross-fertilize or cross-reference those various documents, including our clinical trial government identifier, so that we are quickly able to proceed with FDA approval. We also have the manufacturing data available and approved by the FDA, so we can scale quickly. From a manufacturing point of view, prior manufacturing runs that we conducted at Campbell University's pilot production plant, demonstrated feasibility, reproducibility, and stability. These are important factors in an intravenous sterile drug. These are not trivial matters, and it took quite a lot of work, 2 years to get that accomplished. We can benefit from that in this study now, so it's really critical. We have the CMC section ready to go as well. Plow Center for Sterile Drug Delivery Solutions is making our first batch right now for delivery in July for use in a clinical trial in August. And Particle Science is scaling up so that they could support commercial capacity if required, if the drug proves out. So our investment strategy is show the drug works in the initial patient, show that it's safe in the initial patients, show it works by testing biomarkers initially, and then as we ratchet up the dose, confirm it in a more obvious manner. We're seeking funding in addition from the 2 government agencies that are challenged with coming up with countermeasure strategies to fight this. We anticipate that they will most likely join us in this endeavor when we've started the study and proven the drug is safe in its new formulation. We've raised bridge funding from various sources. We have a new private trust that's committed approximately $250,000 to allow us to manufacture the drug. We need the $1 million to get this phase 1A study started so that we can raise the Series A and B for both COVID-19 and oncology. We have protection for the oncology formulation for the delivery vehicle, as well as a new provisional application for the use of ST-001 in fighting infections. We are really ready to go from that point of view. We have a management team that, as you've seen before in my talks, is really ready to address this from every practical point of view: scientific, commercial, clinical, and so on. So we're going to be there quickly. And then we have the technical board of advisors that provides the same kind of support.
So thank you, Earle. You will notice on the last slide, if you could flip back one, there was Michael Spawn at the bottom there. So I'm just going to summarize very briefly. So the API, the parent compound, Fenretinide, as Earle said, has been in 3,000 patients. It did not have many side effects. Main one was night blindness, was tolerated incredibly well. It's a chemo prevention, so we can give this to normal individuals. Mike Spawn down there, emeritus professor at Dartmouth, was the head of the prevention branch at NCI when I worked there. A magnificent opinion on this matter. He's key on our SAB. IND is approved for CTCL, IP is issued, manufacture is already underway. I have invested some of my own money in that because I believe that the drug is safe, potent, and will rapidly reach an inflection value point at the end of phase 1. Very rapid path to completing the phase 2 in COVID-19. We're aiming by the end of the year. Dual mechanism of action, both anti-inflammatory and antiviral. Short trial path in CTCL. Breast cancer grant submitted, and 12 potential cancer indications. These are our potential exits, collaborations. Obviously, a collaboration with Gilead in terms of remdesivir, potential collaboration with J&J, who owned and developed the drug in the past, and multiple other possibilities. So we see these first as collaborative and then as potential exits. Thank you so much. We'd be very happy to answer any questions.